is Lucia Chemes. And Lucia is expert in the study of structure and function relationship in proteins using both experimental and bioinformatics techniques. She's working in collaboration with national and international groups in Argentina, Germany, USA, and France. Lucia was trained as a biologist in University of Buenos Aires and obtained a Master degree of Science in the Rockefeller University, New York, USA. And she done the PhD degrees from the University of Buenos Aires, and she's currently the leader of a lab at the Institute for Biotechnological Investigations in UNSAM, which is the National University of San Martin. It's a place here in Buenos Aires. So Lucia, I leave you with the people. I stop sharing now. Are you ready? You are muted.
Okay, sorry. Can you hear me now?
Yes, yes.
Okay, and let me know if this is showing full screen.
Yes, it is.
Okay,
nice.
Great. So I'll go ahead. Thank you very much, Chris, for the presentation, and thank you very much for the whole organizing committee for this invitation to give this talk. It's great to be here, and it's been amazing to be hearing the talks during all of these days. So many amazing women doing bioinformatics. So I'm going to try and tell you a bit about the work that we do on studying. I'm focusing a little bit on some evolutionary studies that we've been doing in the lab to understand special kind of proteins, which are intrinsically disordered proteins. As Cristina said, we are working at Universidad Nacional de San Martín in Argentina, and you can see a picture on the right of the institute where we have the group. Oops. Okay, so the first thing I wanted to tell is that our lab is a mixed experimental and computational lab. So the way we try to do the experiments is we do some experimental work by studying protein-protein interactions, by doing structural studies in proteins, and also more recently, proteome-wide screens. And then we also use this data to perform bioinformatic analysis, which feeds us with more working hypotheses to drive the experimental work. So we really try to keep a close collaboration between the two areas in the lab. I'm going to focus, of course, on bioinformatics studies for this talk. So we studied intrinsically disordered proteins, and just to give an overview, the classical paradigm for protein structure and function is that we have a unique structure that determines function, as we can see with active sites and enzymes. And the types of proteins that we study are very different from that. We can see them on the right. They are intrinsically disordered proteins. These are very, very flexible proteins that have equilibria between multiple conformations, as you can see here. And we don't have a unique and defined native state, but we instead have an ensemble of conformations that perform functions. So we're interested in understanding how this happens and how this helps proteins perform their functions in the cells. And we specifically focus a lot on viral proteins, so pathogenic proteins. And proteins from viruses are notably enriched in intrinsically disordered regions. And within these intrinsically disordered regions, there are some functional elements called linear motifs that I'm going to talk about more during my talk, which are very important for viral ability to hijack cell regulation. And we've tried for many years, and we keep trying to understand the role of these functional elements. So this is from a review some time ago where we've been reviewing and studying the role of linear motifs in pathogens. And one very salient feature of these linear motifs, which you can see represented in red in this figure, you can see 4 proteins from different types of pathogens: viral, bacterial, fungal, and even snake venom. And the proteins are represented in green, and the oval that you see in red is an RGD motif. This is a motif for interaction with integrins on the surface of the cell. And we can see that all of these very different kinds of pathogens evolve the same linear motif to be able to interact with receptors and enter the cell. So these kinds of strategies of SLIM mimicry, or evolution of motifs or SLIMs that are able to interact with host counterparts, is a very widespread feature of many types of pathogens. And they evolve convergently, especially because they have small sizes. So we can see a representation of these linear motifs. They are really a linear, they are encoded in a short stretch of amino acids. Here we can see an example of the LXCXC motif. And we can see that both host proteins and viral proteins have these types of elements. And they can have, we can describe them by regular expressions that you can see represented in this box. So leucine or isoleucine in the first position, cysteine in the third position, and aspartic or glutamic acid in the fifth position in this case. But we can see that host and viral proteins that have the same linear motif have very, very different affinities. So we still have a lot of encoding that's going on that is not represented in these regular expressions. So some time ago, we started trying to study these linear motifs in pathogenic proteins. And the first example that we took was the E7 protein from papillomavirus. And we took this example because we had very well-known genotype-phenotype associations in this virus and a lot of natural sequences over which we could do statistics-based analyses. And you can see a representation of the E7 protein from papillomavirus. This is the main oncogenic protein from HPV. So it's the virus causing cervical cancer in humans. And you can see that it has an N-terminal domain that is disordered, and you can see it represented in orange, and then a C-terminal folded domain, and a lot of linear motifs that are distributed along this sequence. So we collected all of these sequences from the E7 phylogeny, and we performed studies on them to see how these linear motifs evolved. So we aligned all of these sequences, and we performed regular expression-based searches to find all of the motifs. And we looked at the distribution of these motifs over the viral phylogeny. And here you can see a viral phylogeny, and the round circles or diamonds represent the linear motifs, and the filled circle is going to be presence of the motif, and empty circle is absence of the motif. So what we could see were that some motifs were much more prevalent than others, and also that there was an uneven distribution along the phylogeny. So some of these viral clades were preserving certain linear motifs, and some of the viral clades were losing them. As we can see here, for example, within this blue clade here, we can see that there's a coincident loss of many of these linear motifs that I'm showing in this example. So the motifs did not seem to be combining randomly, and we tried to study this a little bit more. We reconstructed the evolutionary history of these motifs over the viral phylogeny, and we could see that there were multiple disappearance events of these motifs, which you can see on top. And we also saw that there were multiple independent appearances of the motifs. So here I'm only showing 2 examples, but we studied 7 examples of linear motifs in this system, and we could see that there was a widespread occurrence of disappearances and reappearances of the motifs over the phylogeny, indicating that there was convergent evolution and also many changes of these linear motifs over time. So then we tried to understand whether these changes were somehow related to phenotype in the virus, and we used in this example 2 types of phenotypes. So the tissue tropism, which kinds of tissues the virus could infect, and also the specificity for host. And what we could see by performing statistical analysis using hypergeometric tests in this case was that several motifs, for example, the presence of the E2F motif in this case was correlated statistically to tropism for mucosal epithelia, whereas the presence of the LXSXE motif was correlated to infection of the skin. And also the loss of several of these motifs was correlated to infection of fibroblasts. On the other hand, we also saw that linear motif complement was correlated to the different types of hosts that the virus could infect. So we saw that many motif combinations were linked to specific phenotypic traits. So that was work done a few years ago in collaboration with Juliana Glavina and also with Ignacio Sanchez at University of Buenos Aires. And more recently, Juliana has been doing more of these types of studies on linear motif evolution using the E1A oncoprotein. So this is another oncoprotein, a bit similar to E7 in that it's a very disordered protein, as you can see in this schematic. And it has 11 linear motifs distributed along its sequence. So when we studied this protein, we took a similar approach to the one I described for E7. And we could see, you can see here on the bottom, for all of the motifs that we studied here, you can see 11 different types of linear motifs. We tracked the appearances, the disappearances, and the total number of changes. And you can see that there's quite a large amount of changes in these linear motifs as well in the E1A phylogeny. In some cases, there's more appearances. In some cases, there's more disappearances, and the number of changes also varies, which indicates that these motifs are behaving in a very plastic manner. Some of them are changing a lot. Some of them are changing very little. We also performed association tests like the one you can see on the right to see whether the changes in the linear motifs were correlated between each other. So did two motifs change in a correlated manner? And if you see a black dot, it's because we found a statistically significant correlation between a pair of linear motifs. And we could see that we had a lot of correlations between coincident changes between different types of linear motif pairs. So we found a similar pattern to the one we saw in E7. We have many linear motifs, they are changing a lot, and their changes in many cases are correlated, suggesting they have a functional relevance. But Juliana went on to perform more studies on the E1A phylogeny and try to track how the virus and the host are co-evolving. So in this case, we tried to pair the evolution of the viral tree to that of the host to understand what's going on. And in this case, we could see that the two phylogenetic histories are highly correlated. So there's a high degree of co-evolution of the virus with the host, but the virus does not evolve entirely by co-speciation with the host. So in this case, what we did was try to reconstruct the evolutionary history of the virus, how it adapted to the host, by tracking different types of events that can happen. So the most distributed one was co-speciation. In this case, the parasite co-evolves completely with the host, but there can be other events, such as, for example, a duplication in the parasite over the same host. There can be a host change, so a host switch. So the virus duplicates and one of the lineages switches to a different host. And there can also be partial extinction of the virus phylogeny over the host phylogeny. So what Juliana did was try to reconstruct the history of all of these different changes that have happened during the co-evolution of the viral and the host trees. And we did this by using a heuristic-based approach that provides solutions within a parameter set. So we have to set the cost for each one of these events and then search for solutions that have high support in many events and very few events with low support. So just to really sum up this study in one slide, what we could see in the top left, what you can see is that the main pattern that we observed was that host switch, which you can see in the second line, was correlated with significant changes in several linear motif patterns. So we could see that changes in linear motifs were correlated to an evolutionary event that was the switch from one host to another in the virus. And here on the right, you can see all of these host switch events can be seen in blue. So we can see, for example, that from this lineage here that I'm marking with the pointer, there's a large number of host switch events to other branches of the host phylogeny. And these host switch events are correlated to strong changes in linear motif complement. So the conclusions from these studies about linear motif evolution now, having done a pretty comprehensive study of two viral systems, are that motifs have appeared and disappeared multiple times in papillomavirus and E1A evolution. So these linear motifs can evolve rapidly. There's evolutionary coupling of several motif pairs, so the linear motifs do not seem to evolve independently. The evolution patterns do not depend on the structural context. So the linear motif evolution is motif specific. It's going to be different for each linear motif. And there's a widespread convergent evolution of linear motifs. And there's also an association of changes in linear motifs to viral phenotype, which strongly suggests that these linear motifs participate in viral adaptive evolution. So in the second part of the talk, I want to tell you about another type of evolutionary studies that we've been conducting on viral proteins as well. And in this case, we asked a different question. So we kind of now understand a little bit more about how motifs are evolving. Now we're also interested in knowing how other parts of intrinsically disordered regions evolve. And in this case, the types of regions that we're interested in studying are linker regions. So we took the same E1A protein that I described before, and in color, you can see the part that we studied. So in this case, we studied an interaction, a linker region that joins the E2F motif in blue and the LXCXE motif in red. And both of these motifs mediate binding to the retinoblastoma protein, as you can see in the diagram below. Here's in blue the E2F motif, and you can see the structure. And then we have a linker that joins that motif to the LXCXE motif, which also binds to the retinoblastoma protein. And this was a joint experimental and bioinformatics study that was carried on through a collaboration with multiple groups, but the people in the lab that were doing the work mainly were Nicolas Fautel, who did this as part of his PhD thesis for all of the experimental work, and Juliana for all of the bioinformatic analysis that we performed in this system. So what question did we ask here? We want to understand, can we describe this type of multivalent interaction? So we have a minimal motif, linker motif architecture, and we want to understand how we can describe this disordered linker that we have in E1A and how its properties affect this multivalent affinity for a target. And this would help us explain how the E1A protein is so good at hijacking cell cycle regulation, which is actually what happens when E1A binds to RB. It promotes cell proliferation. So we want to understand this hijack mechanism. So what did we see? I'm going to really be very, very brief with the experimental part. The only part that we did a very large experimental study on this system. We measured affinities and we did a lot of structural work, but I'm only going to show these results, which are the results from Sachs analysis. And just to say that what we could see is that this linker remained highly disordered when it was in complex with RB. So E1A is bound to RB here through the two motifs depicted in red and blue. And we can see that the Sachs data is compatible with a complex where the linker is very disordered and extended and remains disordered when it's bound to its partner. So we decided to see if we could use a model, an entropic model, to treat the linker. So we treated the linker through a model that's called warm light chain model. And in this case, we are describing the chain as an assembly of different conformations that have a particular persistence length and a particular end-to-end distance, complete extension. So how do these models work? In this case, we can see how we would expect the behavior of the linker to be if it agrees with these types of entropic models. And if we think about the system that we have the two binding sites, and we're going to have a first interaction by one of the motifs, and then a second interaction to form the binary complex. And the same can happen on the other side. The red motif can bind first and the blue motif can bind second. So the global affinity in this case, because we don't have interactions of the linker with RB, can be described by the product of the two association rates times the effective concentration of the motif on the second side. So what we can see is when the first motif binds, the second motif is going to have an increased concentration and then bind more effectively to the secondary site. And how can we describe now this effective concentration? We can see in this model that we're using in the diagram below that it's going to depend a bit on the length of the linker. So we have a primary site and a secondary site. And if the length is too short, the second site is not going to reach. If the length, you can see that at the beginning of this diagram showing the dependence of the effective concentration on the linker length. So if the linker is very short, the effective concentration is not high. If the linker has an optimal distance, we have a perfect placing of the second motif on its secondary site, and we have a maximum enhancement of effective concentration, which is the second case. And if the linker is very long, then it explores a wider space, and we again have a decrease in the effective concentration. So we have a range of length that's going to be very optimal for this system. And what we could see is that our E1A system was really poised at the top of this effective concentration curve, and also that our experimental measurements were agreeing very well with this model. So we could use this model to a first approximation to treat the system. And what we saw is that what we get is a very dramatic enhancement of binding affinity from the nanomolar range for each individual motif to a picomolar interaction when both motifs are present, which is the wild type E1A protein. So actually E1A goes in and binds to RB with a very, very high affinity thanks to this tethering mechanism by the disordered linker. And we also saw a very surprising result. We went on to describe this in all of the E1A sequences. So we took the entire set of the E1A phylogeny and we tried to study how this mechanism is evolving. So we take a lot of linkers, and here you can see linkers from 40, 75 residues long. And we did in this case in collaboration with Alex Holhouse and Rohit Papu, we did atomistic simulations of these linkers, and you can see a box plot of the average end-to-end distance of these linkers. And we could see something that was very surprising, which was that we could have almost a doubling in the linker length, but no change in the extension of the linker. So we would expect that a longer linker is going to be more extended than a shorter linker. But actually what we saw is a lot of compensation in this system. So the linkers were variable in length, but their extension, their end-to-end distance, which is actually the important feature for determining the effective concentration, was not changing. The way we would expect here in blue, for example, if we just make a sequence longer without changing the sequence composition, we would expect that as we increase the length, then the end-to-end distance increases. So this told us that the sequence of the linker is compensating with the length in a way that the extension remains constant. And here I'm just going to show a simulation of a short and a long linker, and you can see that the extension of both, it's just for a view, is similar, despite the fact that the linker on the right is much longer than the linker on the left. And we saw that the main compensatory effect was through the net charge per residue. So we tracked a huge number of sequence parameters to see what this compensatory behavior depended on. And we saw that the net charge per residue was the main parameter determining the compensation, in a way that linkers that were shorter, were more extended on average, had a higher net charge per residue than longer linkers, leading to the same end-to-end distance despite changes in length. So what we did was now we used structure-based models. In this case, we used Foldex to predict the binding affinity for all of the motifs in the phylogeny. And we used the warm light chain model to predict the effective concentration for about 100 of these sequences. And then we calculated these global binding affinities to understand how is this high affinity and the tethering evolving in the whole phylogeny. And what we could see in red, it's the system that we studied experimentally. It has a picomolar binding affinity. And we could see that most of the E1A sequences also had a very high binding affinity for E1A, and that this binding affinity for RB, and that this binding affinity was very high and it didn't depend on the length of the linker. So it's a very, very conserved feature in the system. And if we look at the phylogeny, here you can see in one scale that goes from yellow to purple, you can see binding affinity for the E2F motif, for the LXCXE motif, and for the full E1A protein. And in green to yellow, you can see a depiction of the length of the linker. So we looked throughout the whole phylogeny, how are the individual and joint affinities and the length of the linker evolving? And we could see something pretty interesting, which was that in a series of sequences, such as those infecting primates, bats, carnivores, and horses, the motifs were high affinity and the linker had an optimal length. But now if we look here on the right, in gray, it's when we don't have one of the linear motifs, for example, we could see that both things were either highly conserved or lost together. So here in the right, we can see that the sequences either lack one of the motifs, so the tethering is lost, or they have short linkers and low affinity motifs. So this was telling us, yes?
Sorry, you have 5 minutes left, but including question.
Sure, I'm almost done, thanks. So, so what we could see is that the motif affinity and the length are co-evolving in this case. So the conclusions are that these simplified models can help us account for the effect of flexible linkers. They can be approximated as entropic chains, that the tethering can enhance the affinity dramatically, and it can help predict the affinity in multivalent interactions, that despite showing low conservations, the linkers have evolved highly conserved dimensions. So they are under strong selective pressure, and that the linker regions are co-evolving with their associated motifs. So there's a co-evolution of the linear motifs and the linkers that are linking these motifs functionally. And this also seems to be supporting the viral adaptive evolution. So these properties now we know that are important in encoding multivalent interactions in the host and also in the virus. And we recently developed an app that can help calculate these effects of tethering. So this is now in press in Methods in Enzymology. It should be out in a couple of months, but the website is up already. And it allows you to calculate these effective concentrations for flexible linkers. And I also wanted to highlight, because this is a meeting that we're discussing a lot of developments in bioinformatics, that we are part of a community that is trying to help advance the bioinformatic resources to describe intrinsically disordered proteins. And as a part of this, we've done a lot of work on curation in the ELM resource. This is in collaboration with Toby Gibson at EMBL Heidelberg. And then we're also collaborating in other databases such as DISPROT, which is an annotated database of disordered regions in proteins, and also in Protein Ensemble database, which is a database of ensembles of disordered proteins. So we're really trying to do our best to increase the representation of these important regions of proteins as a resource to the whole community. This I'm really not going to talk a lot, but I just wanted to give the link in case somebody is interested. This is a recent work done in collaboration with Toby Gibson's group, where we used these types of analysis of motifs to find new linear motifs in the ACE intracellular tail that helps understand the events that happen during SARS-CoV-2 infection. And just to end, this is our group. I already mentioned the main people who were collaborating, Juliana and Nico, in the work that I described, and on the right, all of the collaborations that we have and the funding. And thank you a lot for your attention.
Thank you very much, Lucia. Oof, such an amount of work and amazing results. Very nice talk. Thank you very much. We have a few minutes for questions, and I'm reading that we have in the Q&A section some questions. Clady Mirela say, congrats for your amazing presentation. My question is, similar conclusions we could say for protein with disordered regions of unicellular eukaryotes?
Well, it's an interesting question. I think we know very little about disorder in unicellular eukaryotes. So the main pathogens, or I mean, we know about it more in multicellular eukaryotes, but definitely it's present, and it's even present in bacteria. So I think it's a great field to work in. There's not a lot known, but I'm sure it's going to increase with time.
Okay, I have another question from YouTube, from Elin Tepa. You know her very well.
Yeah.
And she wants to know, regarding the evolutionary coupling of motifs, how did you check that the coupling is not due to a founder effect?
Maybe she can be a bit more explicit. So what would she consider a founder effect? I mean, one thing that we did, I'm not sure if this is going to respond to the question, but one thing that we did is to make sure that we had an even representation of the sequences in the phylogeny. So for example, we didn't take a lot of subtypes or serotypes that had very, very high sequence identity. So we tried to get one representative sequence from each species so that we don't have a very high overrepresentation. But I'm not sure if that answers the question.
No, I'm not sure about what founder effect is. So we are just on time.